Greetings, my name is Simon Davis, and today I'm going to demonstrate for you how Chinese hamster ovary cells are subcultured in a tissue culture lab. The first step is to remove the flasks from the CO2 incubator and make observations about the flasks. Flask observation will include morphology, contamination, any floaters, miscolored media, and most importantly, the confluence of the flask. Confluency of the flask will determine whether the flasks need to be subcultured or just have their media renewed and returned to the CO2 incubator. Observations we found were flask 3 was confluent, flask 2 was confluent, however flask 1 was not confluent. Before anything is to enter into the laminar flow hood, it must be properly sterilized. This includes spraying yourself with 70% ethanol, as well as spraying a Kim wipe to wipe down any objects with 70% ethanol that are to enter the hood. Our lab assistant has just finished placing in the 3 needed materials for working with subculturing. That includes Ham's F12K media with glutamine, phosphate buffered saline, and trypsin with a concentration of 0.25%. The first step is to remove the old media using a pipette by pipetting out the contents of the flasks from the bottom corner of the flasks. Once pipetted out, the empty depleted media should be disposed of in the container for waste located within the hood. Any disposable materials that have touched the cells need to be disposed of in the biological hazardous waste bin. The next step is to perform a wash on the cells. This is done using phosphate buffered saline, or PBS. PBS is used to remove any unwanted debris still remaining within the flasks. This is done by pipetting out 5 to 10 milliliters per flask and adding it to the flask, then swirling the flask and removing the 5 to 10 milliliters of PBS from the flask. Spills within the tissue culture lab are a common occurrence. They can best be remedied by spraying a Kim wipe with ethanol and wiping in a circular motion inward and disposing of the Kim wipe. Once the cells have been washed with phosphate buffered saline, then the phosphate buffered saline needs to be removed from the flasks and disposed of in the waste containment unit. Since Flask 1 was not confluent, Flask 1 will only receive 10 milliliters of fresh HAMS F12K media and be returned to the CO2 incubator for another 48 hours of incubation. For the 2 flasks that are confluent, they will be split in this demonstration and need to be trypsinized, adding 1 to 2 milliliters of trypsin and incubating the cells in the CO2 incubator at 37 degrees for 5 minutes will cause the cell's adhesion proteins to be cleaved. This effectively removes the cells from sticking to the sides of the walls of the flask. Once the trypsinization step is complete, the flasks need to be diluted with fresh media to halt the process of further cleaving the adhesion proteins. This is done by adding 8 milliliters to complement the 2 milliliters for a total of 10 milliliters. Splitting the 2 T75 flasks into 4 T25 flasks. Since the T25 flasks have a smaller capacity of 3 to 5 milliliters, we will add 2 milliliters of media to each flask, followed by 2 milliliters of cells to each flask. To make the transfer and mixing process a little easier, an intermediate 15 milliliter Falcon tube will be used. Now we will add the 2 milliliters of cells to each of the flasks, respectively. Once the contents of the smaller flasks have been prepared, the flasks will be moved to the CO2 incubator for 24 to 48 hours of CO2 incubation until the next time observations and subculturing needs to be made. Thank you for watching.